Good morning, good afternoon, good evening, everyone, and welcome to the most recent in our series of joint webinar events with SCELSE in Singapore and the Singapore National Biofilms Centre. My name is Mark Richardson, and I'm the CEO of the National Biofilms Innovation Centre, which, as you may know, we aim to be the UK's hub for accessing both capabilities and expertise in the fields of research, innovation and training in biofilms. And we're a partnership of around 60 or more UK universities and 150 companies exploring ways of preventing, detecting, managing and engineering biofilms. And it's a great honour to be involved in this event today with SNBC and SCELSE. As you know, this event is about the emerging area of the microbiome of the air and the built environment and how to investigate and control this. And we've got a hopefully very interesting and what you will find exciting agenda for today, with 4 talks, 2 from the UK and 2 from Singapore. And without further ado, I'd like to hand over to Michelle Birnbaum from the Singapore National Biofilm Centre and SCELSE to say a few words about SCELSE and then to introduce our first speaker. So Michelle, over to you.
Thank you, Mark. Hi, good morning, good afternoon, and good late afternoon here in Singapore. My name is Michelle Birnbaum. I'm the Director of Research Translation and Entrepreneurship, both at NTU and at SCELSE, and as well as running day-to-day activities with Maria Young, Dr. Maria Young at the SNBC, which is the consortium for biofilm and microbiome in Singapore. So just a few words maybe about SCELSE for those of you that don't know the center. SCELSE is the Center of Excellence in APAC for biofilm and microbiome research. And we have over 200 people and quite a nice infrastructure to be able to discover foundational science in biofilm and microbiome and translate that into something that can eventually be used by industry. SNBC, as Mark said, is the Singapore National Biofilm Consortium, and we have industrial partners that range from startups all the way to MNCs, and that work with academia here in Singapore, and again, to foster the ecosystem. And we work quite closely with NBIC in joint events such as this one, but also in looking at different thematics, standards, methods development to further develop the ecosystem on a global basis.
So it's my privilege now to introduce to you the next speaker, who is Dr. Angela Sherry, who is the VC Senior Fellow in the Hub, who is based in the Hub for Biotechnology in the Built Environment at Northumbria University UK. So Angela, over to you to introduce your presentation called Current Challenges of Monitoring the Microbial Quality of Air in the Built Environment.
Oops. So thank you. Good morning, good evening to everybody. And thank you for the invite from the NBIC and the opportunity to give this presentation. Just going to minimize this. I'm Angie Sherry. I'm a senior fellow based at Northumbria University in the northeast of the UK. And I'm affiliated with the Hub for Biotechnology in the Built Environment, which is an £8 million joint strategic investment between both Northumbria University and Newcastle University, funded by Research England's E3 scheme. So our vision is to develop biotechnologies to create a new generation of living buildings, which are responsive to their natural environment, grown using living engineered materials, buildings which are able to metabolize their own waste going forward, reduce pollution. And the reason I'm here today is to discuss how in the future buildings could be able to modulate their microbiome to benefit human health and well-being. So we are a unique blend of researchers. We're a 50-strong team within the Hub. We are architectural and design teams on the Newcastle University side, with biosciences and biotechnology at Northumbria University. And we're coming together in a new transdisciplinary field, which is highly experimental and innovative. And we have expertise in a wide range of disciplines, as you can see here on the right-hand side. So as well as some new labs and infrastructure, so workshops, fabrication labs, we're also just starting to build this unique experimental living house at Newcastle University, which is called the OME. So this is a play on words of some of the methods that we'll use to actually look at the microbiome within the OME. And it's an experimental building which we're going to use to demonstrate some of these lab technologies that we're developing. So we'll be able to test and showcase these at a building scale. So within the hub, we have 4 themes: building metabolism, living construction, responsible interactions. And my role is within the microbial environments team, where, as I've mentioned, we're trying to modulate the microbiome to benefit human and ecological health and well-being. So the first step in this is actually characterizing the microbial diversity and function within the built environment and the relationships of that microbial diversity with different environmental parameters and occupational behaviors and health. So much remains unknown about microorganisms in the built environment and their interactions with humans. We all know that microbes can exert beneficial, neutral, or deleterious effects. And we have this system of ecosystems operating within the built environment, which is depicted by this figure from Prusson and Ma in 2015, where we have influences from the built environment and also interactions with the human microbiome and also with pet microbiome. And within the hub, our aim is to understand the airborne microbiome of the built environment using these high throughput DNA sequencing technologies to determine the key microbial drivers and their function within the environment. We then want to use this data to be able to computationally predict the interactions between the built environment and the different microbiomes that exist. So if you see the figure on the right-hand side, we're going from characterizing the different microbial diversity within the built environment to be able to try and predict from these data sets whether there's a core microbiome that exists between different built environments. So there's yet no definition of what a healthy built environment indoor air microbiome looks like, but presumably it's a diverse array of beneficial and neutral microbes and much less pathogens. And our vision within the hub is to be able to eventually modify or promote and sustain the built environment microbiome in order to benefit human health and well-being. So Stefan's done a really neat job in his presentation of talking about the surveillance of COVID within air samples. And I've just picked up on another sort of allergy, asthma, which affects a lot of the population. And there's evidence that a high bacterial diversity and concentration in built environments has a protective effect. But in contrast, a high concentration of fungi increases the risk of developing asthma, eczema, and allergic rhinitis within the built environment. And in early childhood studies, it's been shown that growing up in a rural or outdoor environment decreases allergy risks compared to children who are growing up in urban environments. And that's thought to be due to the children from the rural environments being exposed to a much richer diversity within their home dust microbiota. So there's some contrasting information about the levels of diversity required to maintain a healthy microbiome within the built environment. And we know going forward that in order to modulate built environment microbiomes, it's not just simply a case of eradicating all of the microbes within a given space. We're not looking for completely sterile surroundings. So we've had a lot of discussions about the indoor-outdoor continuum within the hub as well. So the biological content of air and its movement between the indoors and the outdoors is governed by quite a wide range of factors. So that can be building design, occupant behaviour, ventilation types, differences in building usage and climate. So we know that there's a constant exchange between indoor and outdoor air through natural and mechanical ventilation systems, and also due to cracks in building envelopes as well. A number of studies, as expected, suggest that if you have natural ventilation within a built environment, that the indoor air will mostly resemble that of an outdoor airspace. But much more needs to be known regarding the airborne biological agents and their dispersal into, out of, and around the built environment. And of course, this is a concern more pressing than ever since the COVID-19 pandemic. So I've mentioned a few current challenges so far, as has Stefan in his talk, but reliable microbial sequencing data starts with standardized methods of sample collection and processing. There are choices of different types of air samplers. You can see here on the right-hand side. So you've got samplers which rely on positively and negatively charged membranes to filter the air onto filters. You can also collect without filtrations in these sort of dry cyclonic mechanical air samplers, which is depicted in the very bottom picture. You can also deploy passive samplers, which are just open Petri dishes, for instance, without any agar in them, which will just allow dust and airborne microbes to settle into them. The position of the sampler is important, so it needs to be at a good distance from the floor to avoid over-representing the soil microbiome that may be trailed into the environment on shoes. There are some considerations to be taken into account about how to do your indoor versus outdoor air sample collection. So for your mechanical methods, you're going to require a power supply if they don't run so long off the battery. And then these passive samplers may be quite unstable in adverse weather conditions. And from one of Stefan's team's papers, we know that we're working with very low biomass samples. So in soil, there's approximately 10 to the 12 cells per litre. In water, 10 to the 9 cells per litre. In air, we are 7 to 10 times lower, 10 to the 2 cells per litre, which makes sampling of the air microbiome a significant challenge and requires large volumes of air, which Stefan's already suggested a paper that others can go and look at. So with the mechanical, you have the logistics of runtime versus the number of built environment spaces that you're looking into versus the amount of replication that you require to make your microbiome findings statistically significant. For your passive samplers, what's the best time to leave those in situ? So it's important to talk to like-minded researchers, and we've recently affiliated with the BioAirNet, which is a UKRI-funded indoor-outdoor bioaerosols interface and relationships network. And we can, they provided some access to sampling optimization in the literature, and it's important to have network discussions. At the moment, we're discussing sort of standardization and suggestion of publishing a compendium of methods in this area. So yeah, another challenge is that we need an increase in the number of longitudinal studies that are being performed in order to track microbiome changes in the environment over time. So we're looking for time series data instead of a snapshot of microbiome diversity. So this data generated will add to a wealth of 16S ribosomal RNA gene data that's available for bacteria in these huge databases like the NCBI, mixing it up with the acronym for the Biofilm Innovation Centre. And we need to increase the amount of fungal, viral, eukaryotic and protozoa sequencing data in order to get this microbiome view rather than just focusing in on the bacteria. So we can then overlay the data from the different microbial components to really understand and determine whether there is core indoor microbiomes. In an early sort of classroom study on the air microbiome in 2014, we had quite a short-term longitudinal study where they were sampling 8-hour sampling intervals over 3 days in an outdoor and 2 indoor environments. And as times moved on, we're at a much higher resolution and an increased sort of temporal scale, as we can see from the data in this tropical air microbiome study, where we had 2-hour sampling intervals with 12 time points per day for 5 consecutive days in 3-month intervals over 13 months. It's important to also collect metadata from an environment to look at the total microbiome, because many variables and built environment variables and environmental parameters can influence and drive the microbial abundance and diversity within the built environment. So most studies within the built environment look at occupancy, temperature and relative humidity. So it's important to collect and interpret the genomics and metagenomics data alongside these, the rest of the metadata, and to integrate the microbiological findings with the metadata. So we're using a combination of the MixS Air and MixS Built Environment standard packages, which provide a suggested list of parameters which we should record and report for each sample alongside the microbiology data that we collect from specific environments. And that allows us to then compare the data across different studies. So it's then also imperative to integrate that data from the airborne biological fractions, such as the microbial diversity, composition, relative abundance and absolute cell abundance from cell quantification, and integrate that data with these different environmental parameters which we monitor in the built environment. So also with airflow measurements to identify transmission pathways, with physical measurements of air quality, and also with building usage, occupancy, social behaviours, and with building design as well. And there's a nice review just being published on this in Frontiers in Environmental Science and Engineering. So this will provide a greater understanding of the link between building design, air quality and microbiology, and provide us with the relative importance of different sources of microbes within indoor air. We require going forward more modelling approaches, so computational fluid dynamics and compartmental models, not my expertise, but we should be using those to analyse different ventilation systems and building configurations in the indoor environment, which will help us to then predict and provide early warning signs of any risks to human health within the indoor air. So we've recently started to undertake a study with the Royal Air Force. We're working with general health practitioners and occupational hygienists to investigate the effects of ventilation on the indoor microbiome, including the effects of temperature, humidity and occupancy. We've recently just commenced sampling, which is why this is more of a general overview. Watch this space for the results. We're sampling 11 rooms across 7 buildings, and we're taking an approach of using swabs, passive samplers and microbiological air sampling, which again, Stefan mentioned that they are doing within their studies. And we're longitudinal sampling over 12 months. We're going to use high throughput DNA sequencing and metagenomics to determine the air microbiome, and we'll do this through Northumbria University's Omics DNA sequencing facility, where we have the capability for many different sort of omics technologies, metagenomics as well as metatranscriptomics. We have expertise within the hub to then assist with the bioinformatics and computational biology on the data. And we'll of course then correlate the microbiological findings with the environmental parameters and metadata. And my last slide, the broader reach of the research is to increase the understanding of the benefits of a healthy environment to human health and well-being. Our studies will align with the UK prosperity goals. So due to COVID, we really need to fundamentally rethink our responses to the environment and these social challenges that we face and make transformational changes. So this could come in the form of going forward of some digital sensor technologies, which could create color changing displays, which determine the health of an indoor microbiome. So if you have sensors on the outside of a room, which if they're green indicates a healthy indoor microbiome, and if they're red, perhaps you don't want to enter that room. We want to really make the air microbiome data much more accessible and informative to society. So there's lots of jargon associated with molecular microbiology, and we want to make the data much more accessible. We'd also like to develop novel materials, so maybe taxa-specific antimicrobial surfaces, which we can use in architectural redesign and retrofit of the built environment, which will help to promote and sustain healthy indoor microbiomes. And thank you for listening. Happy to take any questions.
Great, thank you, Angela. We just had the one question come in asking if you will also be looking at old buildings and how to make them better, and perhaps differences between the air microbiome in older properties and newer ones.
Yeah, that's a good question. So yeah, we do talk about retrofit of older buildings within the hub, and the building envelope of older buildings is perhaps more likely to allow the introduction of sort of more microbes within to those microbiome spaces. Other ventilation systems between an old building and a new building might be very different. So yes, we would be interested in looking at the old versus newer buildings.
Our final talk this morning, we're pleased to be able to introduce two UK-based SMEs to talk about some of the challenges they've had in gaining approval to carry out misting to try and control the air microbiome. So it's my pleasure to introduce to you this morning Richard Thomas, who's the Managing Director of Citrox Biosciences Ltd, and also Linden Leadbetter, who's the Director of FogFan Ltd. So Richard and Linden, over to you to introduce your presentation.
So hopefully you can all see this. Thank you, Mark, and thank you very much for the opportunity for us to be able to present our exciting project. So this project actually started over 20 years ago it came out of ICI, where they were looking at how plants protect themselves against air microbiome. And they were looking at how plants did this. And subsequently, we took up the interest in, particularly from a dental point of view, because one of the big problems in dentistry is the aerosol created by high-speed drills. It's been a problem for many years. So we look forward to see how we could provide a natural answer to reducing the air microbiome and therefore reduce the risk of infection. So
sorry to interrupt. Would you like to go onto slide?
Sorry,
yeah.
Sorry. Yeah. Hang on a second. I can't see it. I can't. Where is it gone? Bear with me 1 minute. This stops in the way. Right. Yeah. Sorry.
Top orange bar.
Yeah. Got it. Yeah.
There we go.
Okay. So what we found from the research was that plants use bioflavonoids, and they take natural organic acids from the ground source water, and the combination of these flavonoids and organic acids have very potent antimicrobial benefits. 20 years ago, there was not much written about this, but subsequently there's been quite a lot of research done in the use of flavonoids for their antimicrobial benefits. So what we have in Citrox is a natural combination of organic acids and bioflavonoids, which actually come from a mature bitter orange. And this product serves as a powerful antimicrobial, antiviral, antifungal, particularly good against fungi and yeasts. The product is completely non-toxic, non-corrosive, and as you can imagine, over the last 20 years, we have done extensive testing. One of the issues that we've had is, whereas it's easy to test for its effect on surfaces and surface bacteria, viruses, etc., to actually find a test and a standard to test for air microbiome is actually very hard, and there are very few standards out there, and Dr. Linden will talk a little bit more about that and what we've done in that area. So what we have in the product is a it's not working for some reason. What we have is a very broad spectrum antimicrobial, which is effective against gram-positive, gram-negative yeast molds and fungi. It's also a very powerful antioxidant with anti-inflammatory benefits. It's particularly effective against the cytokine storm, and we have a lot of work we've done in its effectiveness against cytokines. It also is a very powerful effect in breaking down biofilm. We've done a lot of work in the dental area, particularly obviously around biofilm and its effect in the mouth. As I say, it's derived from producing completely natural sources. It's completely biodegradable, safe and eco-friendly. It's easy, it's safe for humans, and everything in the product is what is known as GRAS approved. We are actually doing work in the human health area. We have work currently going on in nebulizing into the air, into the lungs to treat Streptococcus pneumonia. And we've also done a lot of work relating to gut health and its effect on the gut microbiome, which Linden will talk a little bit more about. So, as I say, one of our challenges we've had is we've been able to test this effectively for obviously surface decontamination, but the biggest challenge is how we prove that it's effective in the air against our air microbiome. And because what we believe we have is a product that we can use all the time within indoor environments while people are in those environments, helping to reduce the air microbiome, reducing the load, and therefore reducing the potential for infection. Obviously, you'll never have a completely free environment, and we're not necessarily want that, because obviously as humans, we are, as soon as we go back indoors, we are spreading again, obviously a microbiome. But the whole idea of this is that in a safe way that we can actually reduce the level of microbiome in the air. I'm just going to play you a short video, and then I'll pass over to Dr. Linden.
Keeping your home, place of work, or any indoor space safe can be a challenge, especially during this pandemic. Fortunately, a proven solution is now available that will quickly and efficiently reduce airborne and surface viruses and bacteria. Made from natural ingredients and combined with water for easy misting, our patented Fan Fog Orange is non-toxic, non-carcinogenic, and hypoallergenic, making it safe for people and pets. Fan Fog Orange has been independently tested against multiple viruses and bacteria to prove its effectiveness. Simply dilute Fan Fog Orange with water and add it to one of our easy-to-use fans or humidifiers. Now turn it on and relax as the water vapour and our active ingredients attach to viruses and bacteria and begin to inactivate them in the air and on surfaces. And unlike other potent sanitising solutions, you don't even need to leave the room. Fog fans are suitable for use in any space, including homes, cars, restaurants, shops, medical clinics, offices, or any communal area. Visit www.fog.fan to order yours today.
So Linden, I'll pass on to you and we'll carry on with the presentation.
Hi, thank you, Richard, and thank you for all the other talks. Really interesting this morning. So I'm Dr Linden Leadbetter. I'm a practicing primary care physician in the UK, and I am very interested in gut biome particularly, but also in obviously any treatments we can come up with to improve air hygiene and reduce the spread of coronavirus, hence becoming involved with Richard and Citrox. So when I first came into the project, what I was concerned about was standards. Were there standards? Could we check it for safety? Was it effective? And what we've constantly come up against is the speed of innovation in this area, particularly since SARS-CoV-2, has outpaced the standards and available testing. So there was a paper released by WHO in May 2020, which talked a little bit about surface cleaning via misting. It said it didn't recommend it, it wasn't effective, and it mentioned hypochlorous acid. There was no mention of Citrox or many other products, some of which I presume weren't even available at that time. And if that's the overall standard that's being set to, then if that can't keep up, then how are the governments going to put into place standards to comply with that? That's one of the difficulties here. So what is there, and how did we approach it? So in the UK, there's the Health and Safety Executive. Their broad standard is the control of substances hazardous to health, published in 2002. We came broadly under that. We work with the Health and Safety Executive to make sure we're compliant with everything that they want us to be compliant with, but also knowing that full standard testing isn't currently available. There are some standards. There's the cleaning and cleanliness of ventilation systems, but not the air. There is surface cleaning, that's a new one. And actually we've put forward to have our Citrox tested via EN 17272, and that's surface cleaning via misting. In terms of the air, there is even a standard for non-contaminants in air, but it's compressed air. So again, we're just missing the standards for testing normal breathable air. Next slide, please, Richard. In terms of safety, there's Citrox has been through animal trials. These were limited studies, 10 to 20 mice or guinea pigs, where they checked for acute and shortish term reactions. We went through all of those fine with Citrox. There's no problems there. Human trials, we are starting to look into this area, but this is more reserved for medicines, and we're not using Citrox as a medicine at the moment, so we're not really eligible for these. And then we thought laterally and thought, well, where can we go for studies that would look at breathing in contaminants? So we thought about vapes. So we went to an accredited vape laboratory, Acutus Laboratories in Belfast, UK. It's accredited to ISO 17025. And what they do is they run toxicology monographs on both the raw ingredients and then the ingredients heated to the vape temperature, which is about 180 degrees centigrade. And the results came back very favorable for that. In fact, they said it was the lowest levels of toxins they'd seen in anything they tested, not known to be carcinogenic, mutagenic, or teratogenic. So we did those as well, passed those. But then again, is there an overarching test? No, we're just piecemealing what's available and going forward for everything we can. In terms of aerosol studies and effectiveness, there's only 2 commercial labs we found. There's ARE labs in Kansas, USA, and there's Microchem labs also in the USA. They're commercial laboratories. You can send your product and they'll test it in either a cube or in a room. There are university laboratories. There's some in the UK. There's one in Bristol. They're widespread, but hard to access because they're normally for research purposes rather than commercial. And then the state laboratories or military laboratories, we've got Porton Down in the UK. Again, difficult to access from a commercial point of view. So aerosol testing, we went to one of those US labs. We went to ARE labs. There are limitations with it. You have to decide how you're going to run the test. Every test costs multiple thousands of dollars. And they can't use the real, whatever you want to test, in this case, SARS-CoV-2, they have to use a proxy. MS2 bacteriophage, which is a good proxy, but it's still a proxy. So we sent our testing for them. We went for the 1 meter cube. That was all that was available at the time. And we elected to go for, next slide, please, Richard, 3 minutes of misting our diluted concentrate, so 10 mils of Citrox, also known as Flavorback, and profiled the results over the next 30 minutes. After 5 minutes, there's a 50% reduction in MS2 bacteriophage, and after 30 minutes, there was an 89% reduction, which was great. We'd have liked to have run lots of other mutations and styles of testing, but again, you're just limited by expense. We were happy with those results. We realized that ideally we'd like to have tested against SARS-CoV-2, and other permutations would probably have yielded slightly different results, maybe better. And again, it's in the US, not the UK, so it's just all of this extra layers of difficulty getting these tests done. But they were our results thus far. Next screen, please, Richard. I suppose what makes it really interesting to me, from a physician point of view, is the gut microbe and the fact it's more complex than just killing everything in the air except nitrogen, oxygen, neon. Just there's an air microbiome, as we've heard about today, and we know that some of the other areas of innovation, some of the other products, are looking at killing viruses in different ways, but there's collateral damage with those. So, for instance, we know that exposure to air pollutants affects the gut microbiome. We know that. We know that ozone exposure lowers the diversity in the gut microbiome, not normally a good thing. And it also changes the function of the gut microbiome. So having known all those, we looked at what was available and we thought, well, how would we go about the testing? Where do we see the standards going? So next slide, please, Richard. This is what other people and other companies are innovating. There's simplistic methodologies, so just a HEPA air filter that filters everything above 0.3 microns out of the air. Of course, SARS-CoV-2 is 0.1 micron, so it would slip through that one. There's radical solutions. There's ozone, causes DNA damage if there's humans or animals present. Equally, UVC light, which is filtered by ozone in the atmosphere, causes DNA damage, and the American Lung Association describes it as sunburn for your lungs in a very short period of time. So they seem very radical and are probably taking other things out of the air that we don't necessarily want to take out. And then there's the blanket solution at the bottom there. There's hypochlorous acid, mentioned by WHO. That seems to just deactivate and destroy almost everything in the air. So we thought, or we think, that Citrox has a benefit over all of those in that it's targeted, it's selected. It's not a simplistic answer, it's a complex solution. We know that bioflavonoids positively reconfigure the gut microbiome. We know that what you breathe affects the gut microbiome. We postulate that Citrox will inactivate viruses and microbes that we don't want in the air, but leave ones that we do intact and not have a negative long-term effect on the gut microbiome or on the lung microbiome. So we've sent Citrox for every test we can. It's all come back very positive, but there's more research needed. And I suppose I'd like to just summarize by saying there's been lots of innovation in this area, but we need to be careful about being too simplistic as well in terms of governments regulating it. It's not just about killing the viruses in the air or inactivating microbes. It's about what's left behind in the air and what long-term effect can that have above just the standards, the base layer standards for safety and efficacy. I'll be happy to answer any questions. Thank you very much.
Brilliant, thank you both. We've got a couple of questions that are on a similar vein, so I'll ask them both at the same time. So the first one is, what is your proposed method of application for Citrox in the air? And the second one would be, what are your major target environments for misting with Citrox?
I would have liked to have played the video because actually that's seen. So, Linda, do you want to talk about the application?
Yeah. So the ideal areas would be confined areas like cars, rooms, office spaces, even school rooms, because this is safe to breathe. So you don't have to evacuate the room. You don't have to worry about plants or animals. So anywhere where viruses, especially very small size viruses like SARS-CoV-2, can linger in the air for hours, we think it's ideal in those situations. And the best method for dispersing it is via the Faraday wave system, which is using basically an ultrasonic atomization plate to emit very fine particles of the mist. The finer they are, the longer in the air, the more chance they have to interact with microbes that you don't want to be there.
Brilliant, thanks very much. So any unanswered questions from any of our speakers, we will forward the responses around to all attendees of this meeting. I'd like to thank Richard and Lyndon for a really interesting talk and hand back over to Mark for the wrap-up.
Great, well thank you, Will, and thank you very much to all of our speakers this morning, this afternoon, this evening, wherever your time is locally, on an area that is clearly one of huge interest to people. And what I'm interested in knowing a bit more about, maybe, and I think SNBC and SCELSE are as well, is about whether you'd like to, as an audience, like to hear more about this particular subject matter. So I'd just like to launch a poll, and I'd be very grateful if you were just willing to have a look at this and give your answer to this, to see, would you like to see further webinars on this general topic of the air and the built environment microbiome, collaboratively between ourselves and SCELSE-SNBC, particularly contrasting perhaps these two different environments we have with the tropical and the, if you like, the more northern hemispheric, and some of the challenges, as we've heard about today, in regulating and controlling this. So I'd be interested to know if there is adequate interest. We will certainly schedule further matters on this area, and if you're interested in participating in those and talking or presenting your work or your thoughts, then please do let us know, either at SCELSE or at NBIC by our usual routes, that you're interested in participating. And I'll leave that poll open while we finish, but I'd just like to perhaps complete by saying thank you very much to all of our speakers for their excellent presentations and for launching us into this field. Also to our audience, you, for attending and showing such engagement with this topic. And any questions that you've asked that we've been unable to answer, we will make sure the participants get to see those and you receive the answers. Okay, so without further ado, I'd just like to hand over to Michelle to perhaps say farewell on behalf of SCELSE-SNBC, sure, and then I will close the webinar.
Yeah, I'd like to also reiterate what Mark just said in terms of the presentations. All the presentations were really interesting and raised a number of interesting points, actually. And I think that hopefully the polls will say that we should get more into depth into having more webinars on the air microbiome, and especially between the built environment and the outdoor air sampling, as well as the difference between tropical and more temperate climates. Those are always interesting differences, and they bring up some interesting points. So I'd like to thank everybody, including our audience, both abroad and in Singapore, because it's almost dinner time here, and people are still around.
Thank you.
So kudos to everybody, and thank you to NBIC. We always love working with you guys. It's always a very good partnership. So thank you very much.
Thank you. Thank you very much. And thank you, everyone, and have a good evening, a good afternoon, or a good morning. Thank you.
Bye-bye.